So the case of Aedes albopictus is a perfect example of how global change can introduce new invasive species and pathogens, and global change is increasing this presence of invasive species by climate warming, urbanization, and human mobility specifically. And the introducement of these new vectors and parasites can help new disease outbreaks to occur, since the new hosts can be found as well as ideal environmental conditions. So regarding the case of pollutants, there's a long list of pollutants present in nature, and one of these pollutants are antibiotics. So antibiotics are present in freshwater ecosystems worldwide, and since larvae develop mostly in these freshwater ecosystems, the presence of antibiotics may affect the mosquito microbiota and therefore affect their vectorial capacity. So now I'm presenting you the conceptual framework of the thesis. As I said, we are studying these 4 global change components and how they affect different aspects of mosquito biology, specifically survival, body size, immune system, gene expression, parasite interaction, and of course microbiota composition. So in this case, I wanted to place the microbiota as the center of the framework, since it regulates the interaction between hosts, vectors, and parasites, and at the same time is affected by the environment and specifically by global change components. Talking a bit more about the microbiota, it is mainly acquired during the larval phase and maintained until the mosquito reaches adulthood, although some tags are lost and others are gained throughout the process because of mosquito feeding behavior. Here I present you vectorial capacity, which is, describes the ability of a population of vectors to transmit pathogens to a host. And traditionally, studies have focused on these components of the vectorial capacity, such as vector density, vector biting rate, competence, incubation period, or survival. However, microbiota seems to be a key component altering all of these other components of the vectorial capacity, and still little attention has been given at how global change affects the composition of the microbiota. So as I said, there's an interaction between microbiota and pathogens, and this can happen either directly or indirectly. Directly, the microbiota can inhibit pathogen growth or compete with the pathogen for resources. And indirectly, it can stimulate the mosquito's immune system, making it hard for the parasite to establish. Here I'm presenting you the model species used in our project. In this case, Culex pipiens is a native mosquito, the common mosquito. It shows ornithophilic feeding behavior and can transmit West Nile virus, Dirofilaria, and Avian malaria, among others. And then we have Culex perexigus, which is quite similar, although its habitat is quite different, and is the main vector for West Nile virus here in Spain. Then Aedes albopictus, as I said, is an invasive species, native to Southeastern Asia, and interestingly, its feeding behavior, it's mammalophilic, and it is diurnal, which makes it a perfect vector for the transmission of dengue, Zika, chikungunya, yellow fever, and at the same time really annoying to people. So there are already some published studies about the mosquito microbiota of this species. In this case, this study found that Wolbachia dominated the microbiota of Culex pipiens and Aedes albopictus. However, in the case of Culex etalongia reolata, there seems to be a different relative abundance of the bacteria. And interestingly, in the case of Culex perexigus, there was a virtual absence of Wolbachia, and this might have some implications in disease transmission. Moving on to the objectives, the main aim of the thesis is to investigate how global change affects the composition of mosquito microbiota and pathogen interaction. For this purpose, we established 3 different objectives that I will be talking about later on. Objective 1 will be assessed in chapters 1, 2, and 3, objective 2 in chapters 4, 5, and 6, and objective 3 in chapter 7. Regarding objective 1, our aim was to identify environmental factors determining the mosquito microbiota composition and pathogen prevalence under natural conditions. So specifically, we wanted to investigate how temperature and urbanization both influence the microbiota and prevalence of pathogens such as avian malaria and West Nile virus in these 3 species. Objective 2 is more of an experimental approach where we combine the effects of temperature and antibiotic exposure on several aspects of mosquito biology that I introduced you about in the conceptual framework. And objective 3 is another experimental approach, also studying the combined effects of temperature and antibiotic exposure, in this case on pathogen infection. So the mosquitoes from the prior experiment will be exposed to infected and uninfected house sparrows by avian malaria to test the effects on mosquito microbiota. Regarding the methodology, mosquitoes were captured in the field. In the case of objective 1, mosquitoes were captured as adults with EBD Sentinel-2 traps in Seville and Granada. And for objectives 2 and 3, mosquitoes were captured as larvae for experimental rearing. So regarding this experimental rearing, mosquitoes were maintained in climatic chambers at 25 and 30 degrees and with water containing antibiotics or without them. For pathogen detection, we use PCR techniques to identify avian malaria parasites and West Nile virus if possible. Moving on to the microbiota analysis, DNA extraction was done in the mosquito pools, and then RNA-16S RNA sequencing, read processing, taxonomic assignment, and diversity and richness analysis. And for the transcriptomic analysis, gene expression was analyzed after RNA sequencing and read mapping. Now we'll talk to you about each of the chapters in the thesis. So chapter 1 is entitled Urbanization and Vector Species Shape Avian Malaria Prevalence in Mosquitoes. This chapter is already published in Integrative Zoology. So mosquitoes were captured during 2 years in 3 urban and 3 peri-urban sites from the city of Granada, specifically Culex pipiens and Aedes albopictus. And our results showed that all positive samples belonged to Culex pipiens pools from peri-urban sites. And we obtained 6 different Plasmodium lineages with 2 co-infected pools. In the case of Aedes albopictus, despite being an invasive species and commonly thought to transmit many parasites, in this case, it was Culex pipiens, the native mosquito, which showed a 2.53% prevalence of avian malaria parasites. Moving on to chapter 2, it studies the impacts of urbanization, species, and parasite infection on mosquito microbiota composition. To do so, mosquitoes from the first chapter were used to compare the microbiota composition according to mosquito species, habitat type, and infection status. And here I'm showing you some of my very preliminary results. Here we are comparing both species and both types of habitats, and at first glance what you can see is some differences specifically in Culex pipiens from urban sites, but this may be biased because of the low numbers of Culex pipiens found in these urban sites. And you can see that Wolbachia also dominated the microbiota of both species. So chapter 3, we want to test how environmental temperature shapes mosquito microbiota composition. To do so, we conducted a mosquito collection in 5 sampling sites from Sevilla from June to November, in total 13 sampling sessions. And we analyzed Culex pipiens and Culex perexicus pools to test the composition of the microbiota and parasite prevalence. And in each sampling site and session, temperature and humidity was recorded using data loggers. So chapter 4 is one of the main chapters of the thesis. Here we wanted to test the effects of temperature and antibiotic exposure on different components, such as immune status of the mosquito, survival, and body size. To do this, we conducted microinjections to extract the hemolymph of the mosquitoes at 3 different ages, with the objective of counting immune cells. Those are the cells that you can see in the picture. And here we have some of our preliminary results. They suggest that antibiotic exposure and temperature both affect the number of hemocytes, with contrasting effects depending on mosquito age. So as you can see, particularly the main differences are found in the case of 10-day-old mosquitoes. And there's a similar dynamic between the control at 25 degrees and the antibiotic at 30 degrees that we are still trying to understand. For the survival, female mosquitoes were monitored over 31 days to calculate survival rates. These results are based on over 10,000 female mosquitoes, and in the graph you can see the 4 treatments. As you can see, control and antibiotic groups show a similar dynamic, but in the case of temperatures we can really see some differences. So we can see higher mortality at 30 degrees in comparison to 25 degrees mosquitoes. Regarding body size, wing size is used as an estimation of this body size, and what we obtain is that mosquitoes showed shorter wings at higher temperatures. In this case, chapter 5, we aim to identify the antibiotic resistant genes in control and antibiotic treated mosquitoes. To do so, we extracted the DNA with a specific kit and then used qPCR to identify these resistant genes. And we found these 4 resistant genes with different functions and under different experimental conditions. And we are currently working actively on this, trying to understand how this works. Regarding chapter 6, it consists on an integrative analysis of the mosquito microbiota and gene expression. What we have already done is extract the mosquitoes from each treatment at 3 different ages, and these mosquitoes are frozen, and we will soon start processing them. And finally, chapter 7, here we want to test the interactive effects of temperature, microbiota disruption, and pathogen exposure, specifically on mosquito microbiota dynamics. To do so, mosquitoes will be reared as in the previous experiment, and birds will be captured in the field. Then these mosquitoes will be exposed to these birds, and we will test the composition of the microbiota between treatments and between infection status. This experiment is expected to start September 2026, probably. So we have 2 potential options for the research stay. The first one could be at the University of Uppsala in Sweden. It would be more of an in-depth characterization of the microbiota using sequencing, bioinformatic approaches, more of an in-detail analysis of the microbiota. And option 2 could be conducted at the Bernard Natsch Institute for Tropical Medicine in Germany to experimentally assess the effect of antibiotic treatment on the development of West Nile virus in mosquitoes. So my PhD committee meeting was conducted on March 30. We discussed the timeline of the thesis, the structure. I received recommendations, suggestions, and I was able to receive really positive external feedback. So overall, it was a really nice experience. I would recommend it if you can. And finally, the timeline of the thesis. So regarding chapter 1, it's already completed and published. I am working actively on chapters 2 and 3 on the analysis. I'm writing the manuscript for chapters 4 and 5, and chapters 6 and 7 will be conducted during the last 2 years of the thesis, although I have some progress in chapter 6. The research stay potentially could take place in the year 2027, but this may need any adjustments. So I want to thank my team for all the work and support in this project, and of course to all of my other mosquito colleagues and all of you for listening. Thank you very much.
Okay, thank you, Paula. Great talk and amazing project. Now, keep your questions because we'll have a second talk now from another student, and we'll have questions for both of them in the end, okay?
Yeah, I think I can use this. Okay. Thank you.
Yeah, I can introduce William. So William is coming from the Londrina University in Brazil, and he's doing the PhD with Claudia, who is the main supervisor there in Brazil, and with me. So he came in February for a stay. I wouldn't say a short stay because he's living in by the end of July. He's doing some complementary experiments to the main experiments that he's going to be talking about that belong to the Williams thesis. So yeah.
Okay. So can everybody hear me? All right. So good afternoon, everyone. As Pablo said, my name is William. I'm a PhD candidate at State University of Londrina in Brazil, and I'm currently researching 2 main stressors, human-driven stressors in amphibians, which is climate change and chemical pollution. So this work is supervised in Brazil by Dr. Claudia Martinez, and here in Spain by Dr. Pablo Buraco. Okay, I'm gonna first briefly talk about my academic background. This is the beautiful campus of my university back in Londrina, where I got my bachelor's degree, and I discovered ecotoxicology, which is the field I currently work in. Later, I did a master's on nanogeraniol effects on amphibians, and in 2024, I started my PhD in the university, which is I'm going to talk about here today. In 2025, I published 2 papers from my master's work, and from February until July, as Pablo said, I'm doing the research stay here at Doñana. So the main line of questions of my career is how human stressors affect amphibians throughout life. And to set the stage, I want to talk about 2 main stressors. First, starting by climate change. I'm sure most of you all know that climate change causes ecosystem disruption, which leads to habitat shifts and biodiversity loss. This is true for a pond-breeding amphibian where warmer temperatures could cause favoring the pathogens and causes disease that could kill populations of amphibians. Also, they need the moisture-rich habitat to complete their life cycles, and heat waves could dry up these ponds. As well as shifting seasons could affect their breeding timing and the larval development time. That's why amphibians are considered sentinels and highly vulnerable to climate change, because they are biphasic, they need the water, and because of all the other things that I've talked about before. Okay, the next stressor I want to talk about is conventionally synthetic pesticides that bring familiar problems we already know. They have highly non-target toxicity. They need to be applied frequently in the field to do their action, and also they leave harmful residues in the environment. Thus, an alternative to them are essential oils. They are natural, they are biodegradable, and they have established target toxicity, but we still don't know yet their non-target toxicity. One of the main flaws of essential oils is that they rapidly evaporate, so they are highly volatile, and thus they need to be reapplied frequently on the field, which could lead to high concentrations on the environment. Thinking about that, our collaborators from the NanoAgro network developed a method to encapsulate these essential oils in a protein extracted from corn, and thus getting a nano-biopesticide. When you apply this nano-biopesticide, the encapsulation provides a controlled release of the essential oil, so you don't need to apply it frequently because it's going to get released throughout time. Also, it provides long-lasting protection, enhanced efficacy, and thus needing less times of application. So it's considered a sustainable and effective green pesticide. Okay, so the overarching question of my thesis is how does climate change, specifically through temperature rising, alter the susceptibility of amphibians to nanoparticles? And I'm going to answer that through 4 chapters. The first chapter asks the same question, but in the scope of the whole literature. So, are green biopesticides safe for non-target species? To answer that, we chose 2 stressors, essential oil effects and nanoparticle effects. And we're currently, we already defined the team, built and validated the search string, and run them to Web of Science, Scopus, PubMed, and CAB Abstract to get roughly about 1,500 papers from 2016 to 2026. And we're currently at the screening criteria, applying the inclusion and exclusion criteria. Okay? Moving on to chapters 2 and 3, they will basically ask the same questions. Can temperature rises predicted by the latest IPCC scenario alter the susceptibility of bullfrog tadpoles to eugenol or thymol-loaded nanoparticles. They are two different chapters. They're twin chapters. So they will have the same design, but for different compounds. To answer that, we sourced the tadpoles from a frog farm in Brazil, and they were all at larval stages. And they were led to the laboratory where they stayed for 7 days for acclimatization in 3 separate rooms with 3 different temperatures up to a warm of 4 degrees. The other water parameters stayed basically the same in every other room. It's worth to note that they had a 14 to 10 photo period, ad libitum feed, and 80% water renewal was done every other day, and the parameters of the water were tracked with a multi-parameter reader. Then after acclimatization, the experiment lasted for 7 more days, ending on day 14 with tissue sampling. So for eugenol, we had 5 treatments spread across 3 temperatures. So we had a water control, a solvent control, an empty nanocapsule control, the free eugenol, and nano-encapsulated eugenol, all at 4 milligrams per liter. And the same is mirrored for thymol. So control, solvent, empty nanocapsule, thymol, and nanothymol at 2 milligrams per liter. So these concentrations correspond to roughly a dilution of what is applied in the field. Okay, after the experiments, the animals were submitted to the same procedure. First, we anesthetized them and got their weight, length, and staging. After, we got their blood by cardiac puncture and spun down to plasma. Later, we euthanized them and got their tissues, which were stored at -80 until analysis. And on the different tissues, we assessed different biomarkers. So we assessed genotoxicity through alkaline comet assay, immunotoxicity through white blood profile, white blood cells profile, neurotoxicity through acetylcholinesterase, biotransformation by EROD and GST enzymes, osmoregulation through plasmatic ions and the Gill pumps, energy metabolism through hemoglobin, glucose, lactate, and glycogen content, and also the battery of oxidative stress through reduced glutathione, catalase, superoxide dismutase, and also lipoperoxidation. Okay, because all these data were coming from different tissues and different conditions, we had to have a multivariate approach to analyze all of this. So for that, we auto-scaled the data through the z-score, and then we performed a Permanova multivariational test to assess the multivariate variance of biomarkers through temperature, treatment, and their interaction. We also did a PermDisp test to check for homogeneity. Later, we did a partial least square discriminant analysis to see the separation of the treatments. And the VIP scores will tell us which biomarker is driving this separation that we see in the PLSDA. And finally, we have a cross-validation score based on error balance just to tell us if the model is actually being predictive or not. So, beginning with the results with eugenol. So we can see that all the factors and the interaction were significant. So, roughly temperature and treatment are explain about 7% of the variation, with the highest effect being of the interaction for eugenol. Thymol changes this balance, so the highest effect was from the temperature. The treatment remained at around 7%, and the interaction was still significant, but with about 13% of the variation of the data. This is important to notice that PermDisp flagged the temperature from eugenol and thymol as unequal, which means that the animals were responding in a heterogeneous way. Okay, For the next results, I'm going to guide you through how to interpret the graphs. You're going to see that the points represent the individuals, and the ellipses will represent 95% of confident ellipses. The VIP scores of the bars will represent each biomarker that drive the separation, and VIPs above 1 represent a confident biomarker, relevant one. And the k-fold cross-validation is based on the error rate. So the lower the cross-validation, the better the model. So beginning for the temperature in eugenol, we can see that moving on for the first axis, moving on from right to left, left to right, I mean, you get it, right? So from the red one to the blue one, let's say that. From the warmest temperature to the coolest one, we have a difference. But because the data was very heterogeneous, we can see that it is heavily overlapping. So it's a genuine but modest model. By treatment now, in the coolest temperature near comfort for these animals, we can see that the treatments all overlap. And what little difference there is led by glucose and muscle biomarkers. At the intermediate temperature, though, we can see that the controls begin to diverge from the treatment groups. And this diversion is driven by gill biomarkers. On the warmest temperature, we can see the cleanest separation. And again, glucose and muscle markers are driving this separation here. It's also the best model because it has the lowest error rate, sorry. Okay, moving on to TIMO now, we can see that the temperature effect here is stronger than the eugenol one, and we can see that the warmest temperature is very separate from the coolest ones. This is a high predictive power because it's only 12% of error rate. On the coolest treatment, though, again, near thermal comfort, we have overlapping of the treatments. So the animals don't seem to differentiate the treatments that well on the coolest temperature. On the intermediate temperature, we can see that nanoTIMO and free TIMO begin to separate from the controls, driven by the oxidative stress markers. At the warmest temperature, we have the best separation, with nanoTIMO being completely separate from all the controls, and also the free TIMO coming to this part. And this is led by the brain biomarkers. So the brain is being most affected in the warmest temperature in TIMO exposure. So as a synthesis of the both chapter, we see that the both temperature and treatments for eugenol have moderate effect, and the interaction is the highest one. We can also note that there is no solvent effect because it was always close to the control in the graphs that I showed you before. So we can say that the strongest formulation effect is nano-eugenol at highest temperature. And the key axis driving it is energy metabolism and neurotoxicity. For TIMO, it shifts. The dominant effect now is temperature. Treatment has a weaker effect, and the interaction has a moderate one. It's the same here for the solvent. It has no effect. And it's a very good predictive model because of the lower error rate. The stronger formulation effects being nano-TIMO at the highest temperature, driven by biotransformation and oxidative stress battery. So the takeaway message is that these nanoformulations are not inert, and as the climate warms, they amplify their distinctiveness in the animals. Okay, you're following me? All right. So the next chapter is a final chapter derived from the previous one. And because these compounds, TIMO and eugenol, are applied in vineyards together, they are likely to co-occur in the environment together, okay? So thinking about it, we ask how does the eugenol plus TIMO mixture, free and nano-encapsulated, alter the bullfrog physiology from larvae to juveniles? For that, we're going to have three treatments and a control. And we're gonna sample the individuals at 2 ontogenetic stages, the first being at larvae, and then we're gonna let them grow into juveniles and sample them and run all the biomarker panels that I've told you before again. So where I'm at the thesis now. Chapter 1 is roughly 5% complete because we are still deciding on stuff in the screening criteria, so we have a lot of things to do regarding the systematic reveal. Chapters 2 and 3 are basically 80% done because I've run all the analysis and I'm writing, and I just need to publish, basically. And then chapter 4, I haven't started yet. I'm going to start it when I come back to Brazil later in August. So that leaves me in the midpoint of my thesis. That I'm the end of the second year. I still have 2 more years to go, so I'm in the middle of the thesis. Which leads me to my research here at EBD with Pablo de Olavide since February, that I've been working in redox analysis with a previous done experiment in the lab. I'm also collaborating in the Allen experiment, artificial light at night, experiment led by Troy, and I'm also conducting an experiment on my own of post-light pollution regarding speed camera flashes during periods of high and low ticketing here in Spain. This is the Allen team, and through this stay, I could broaden my ecological view because I feel like I had more of a molecular view coming from ecotox back in Brazil, and now I have like the whole big picture, not the complete picture, but I'm trying to work on that. And I could learn new lab and field techniques and also establish an international collaboration that I hope to carry forward. So I want to thank my university and my graduate program, also the Estación Biológica de Doñana for receiving me, and CAPES Foundation because I have received the grant for coming here through this institution. I also want to thank Pablo very much for receiving me. He promptly accepted me through email. We haven't even met each other before, and I was like, can I come? And he was like, yes, you can come. So yeah, I want to thank you for that and for all the supervising orientation process. Oh. Okay, and also last but not least, I want to thank my laboratory in Brazil. My supervisor, Claudia Martinez, is a brilliant researcher in the ecotoxicology field. She adds so much to the ecotoxicology field, and I'm so inspired by her. I also want to thank all my lab mates for doing all the work with me, basically, because without them, I couldn't complete all those experiments, which was large experiments for my lab. Okay, that's it. Thank you. If you have any questions and suggestions, please.
Thank you very much for your talk. And if you have questions now, you can. And for William and Paula talk, so you can come as well if you want. So is there a question to ask?
So thank you very much to both presenters. Very nice talks. And my question is for Paula. So you mentioned that one of the targets of your PhD is identifying and characterizing genes related to antibiotic resistance.
Yeah.
And my naive question is, are those genes in the mosquito genome or in the bacteria?
In the bacteria.
Okay,
Yeah.
because I was starting to think some kind of horizontal transfer which may occur eventually.
Well, it may occur. One of the genes that I showed you that we identified, it's a typical horizontal transfer gene. But yeah, in the microbiota, these genes that I'm talking about.
Okay, but you are focusing on the genes in the bacteria.
Yeah, exactly. Yeah.
Okay, thank you very much.
Other question?
So do you know, I mean, you've been talking about neurons, amphibians. Do you know about these effects of these kind of essential oils on other fauna, like maybe insects, and whether they are maybe spill over to other animals or something like that? Is it something known?
Okay, thank you for your question. So it's about effects on other fauna and mainly on insects, right? So this is very established because they are being developed to act as pesticides. So one of the main pests are insects, so they have insecticide properties. Mainly the one that I researched in my master's, which is geraniol. It has insecticidal effect. Eugenol is more used as an anesthetic, and thymol also has insecticidal effects. And this is their target toxicity. As non-target toxicity, I would say it is more researched in mammals, mainly rodents.
Mm-hmm. Yeah. All right. So maybe those effects on amphibians, they are things because there is some kind of leaching, or when they are applied in crops and things like that, or I mean, are they used to control anything that is in the aquatic, bodies, or?
No, I wouldn't say they are used to control anything in aquatic bodies. So they are used as pesticides in, for example, the vineyards I told you, which they are sprayed, and then if you have rain or leakage, it could get into the water environment.
Yeah.
So they are not designed to be used in water, but they could end up in water. That's why we say of non-target toxicity. For amphibians, fish, or whatever, it's in water, it's not the target.
Yeah, no, that's very interesting, because we always think that natural substances are good, but they can be as harmful sometimes as some other artificial, chemicals, I guess. So
yes, definitely. Especially in the context of warming, that I've just showed you, that the toxicity changes as climate warms. So you cannot deem one thing as safe now that it's not that hot. If it's gonna get even warmer, is it still gonna be safe? And we're seeing that it's probably not.
Yeah, that's very interesting. Thank you very much.
Thank you.
Thank you. Is there other question?
I have a question for each. So, William, how does the effect that you saw compare to the effect of pesticide? Are they of the same scale, or is it a lot less?
So thank you for your question. The effects are the same between pesticide and non-target. That's your question, right? So I wouldn't say they're the same because, well, I have to think a little bit. But sometimes we see, I'm talking about regular pesticides now because we don't have much data on biopesticides, but sometimes we see, for example, that a pesticide exerts neurotoxicity to acetylcholinesterase inhibition, right? And the insects, they lead to cholinergic how do I say that in English? Like a cholinergic stress, let's say. And when we see their non-target toxicity, it's the opposite. So instead of inhibiting the enzyme, they make it more active. So I wouldn't say they have the same effects for targets and non-targets.
But I actually meant, do they have the same effect as the essential oil? Like for the frog, is it worse to be exposed to the oil or to the pesticides?
The nanopesticides, you mean? Or the regular pesticides?
The regular ones.
Ah, okay, like a comparable approach. Okay, now I get it. Sorry. So, well, I don't know for sure. We have to have a balance, you know. Maybe it would be a good idea to conduct an experiment actually testing that. Yeah, I wouldn't know how to answer.
Yeah, thank you. Thanks.
Thank you.
So yeah, I just had the question about the age. You saw a different effect at 0, 10, and 20 days. I was wondering what that age mean for the mosquitoes. So do they die at 20 days or?
No, they can keep on living. Like some of them, I had to kill them at like 100 days because they were still living. So yeah, day one is like a basal to test the basal capacity of the mosquito immune system or whatever. Day 10 seems to be like the time the pathogens takes to develop inside the mosquito. That's why we chose day 10, because it seems to be like a important physiological stage. And day 20, the parasite would have already like been there for a long time, and the mosquito is in an advanced age. So that's why we chose those 3 life stages.
Yeah, thanks.
Thank you. Other question online as well? I don't know. No more questions? So thank you very much for your talk, and thanks to all for coming, and we will see you next week for the next seminar. Thank you.
Thank you.